Hello everyone. So the next topic which we will be studying is about the Calvin cycle. This cycle is also known as the C3 cycle and it is also known as the carbon fixation cycle. So these are the different names of the cycle which is being represented in the slides also. So what we will be discussing is about what is Calvin cycle, how the process of Calvin cycle is taken place in the plants, then what are the three phases of the Calvin cycle, what was the history of the Calvin cycle, and how we can control the Calvin cycle. So let's start with the first slide of the Calvin cycle, which is the introduction slide. Calvin cycle basically is present in all the oxygenic photosynthetic organism, that is plants. C2 reduction in fixation occurs through the carbon cycle, that is carbon fixation cycle, which is also called as the Calvin cycle. So the Calvin cycle is a series of biochemical reactions which reduce CO2 to 3 carbon sugar. So it is being converted to 3 carbon sugar, that is why it is also known as C3 cycle. In eukaryotes, it takes place in the stroma of the chloroplast, and in case of the prokaryotes, it occurs in the cytosol. So this is the introduction part, that in eukaryotes it takes place in the stroma of chloroplast, and in case of prokaryotes it occurs in the cytosol. The next is about the history. So the Calvin cycle, it was being elucidated by the Melvin Calvin and the Andy Benson at the University of California, Berkeley, for which they got a Nobel Prize in 1961. So for this cycle, they got a Nobel Prize. Then what are the three phases of the Calvin cycle? The reactions of the Calvin cycle can be divided into three phases, that is carboxylation, reduction, and regeneration of the RuBP. We will understand this RuBP in detail in the carboxylation slide only. So, carbon fixation. The first stage involves the incorporating carbon from carbon dioxide into an organic molecule. So here RuBP, the full form of RuBP is here, that ribulose 1,5-bis phosphate, which is a 5 carbon acceptor molecule. So the carbon dioxide combines to this RuBP, and an enzyme called RuBP carboxylase or oxygenase or RuBisCO catalase, attachment of carbon dioxide to RuBP. So this process make a 6 carbon compound. So earlier it was a 5 carbon compound, so when an enzyme acts on this, they will make a 6 carbon compound. The compound is very unstable, so it would split into 2 to 3 carbon molecules compounds, which is 3-phosphoglyceric acid. Then hence for each of the carbon dioxide that would enter the cycle, there will be 3 PGA molecules which are formed. We will be discussing all these in the cycle in detail also. The next step which comes is the reduction step. So in case of reduction, this is the next phase. So the reduction stage requires ATP and NADPH. These compounds are used to convert the 3 PGA molecules into a 3 carbon sugar, which is known as glyceraldehyde 3-phosphate or G3P. In the first step, each molecule of 3 PGA would receive phosphate group from the ATP, and which is turning into 1,3-bisphosphoglycerate, which is a double phosphorylated molecule. So this would leave the ADP as a byproduct. So the second step which comes into this picture is the 1,3-bisphosphoglycerate molecule. They are reduced by gaining the electrons. So that is being reduced by gaining the electrons. So each of the molecule would receive 2 electrons from NADH, and they loses 1 of its phosphate group. So after which the glyceraldehyde 3-phosphate or G3P, a 3 carbon compound is being produced. So in the second stage of reduction, it produces phosphate and NAD plus by as the byproducts. So this was about the reduction stage. Then comes the regeneration stage. What happens is regeneration is some G3P molecules, they produce glucose while the others would recycle in order to generate the RuBP acceptors. So RuBP, in the first slide of carboxylation we have seen that what is RuBP acceptors. Then this stage would require ATP. So this stage would require the energy, and they involve a very complex set of reactions in this. So this the regeneration stage has been there. So the three molecules of carbon dioxide, they enter the cycle. So in order for one G3P molecule to leave the cycle and on to the glucose synthesis, and they provide the three new atoms of fixed carbon. So there are 6 G3P molecules which will be produced when the 3 carbon molecules will enter the cycle. So the cycle will be represented covering all these steps in detail. So one would leave the cycle to be used to produce glucose, while the rest would be recycled in order to regenerate 3 molecules of the RuBP acceptors. This is about the Calvin cycle. So here it is a picture which is being depicted here, that how a C3 or a Calvin cycle is being shown. So there are basically 4 steps in the Calvin cycle. So first step is carbon fixation. We have discussed the carbon fixation. It requires 6 CO2 molecules which will convert 12 molecules of 3-PGA, that is phosphoglycerate. The 12 ATP will be required in order to produce 12 ADP, and then the 12 NADPH molecules will be required to convert the 12 NAD plus ions. Then it will convert into 12 G3P. Then this was about the second step. Then about the third step. In the third step, what is happening? RuBisCO molecule, that is 2G3P, is transported from the chloroplast to make the glucose, which gets converted into 6-ribulose 5-phosphate, and after this they will require the ATP for this energy, and they will release the 6 ADP molecules. This is the fourth step which we are talking about, and 6 ribulose 1,5-bisphosphate molecules are being produced here. So this is about the Calvin cycle, in which carboxylation is the fixation of the carbon by the carboxylation of RuBP. Reduction is the reduction of the fixed carbon to begin the synthesis of triose phosphate, and regeneration, it is the synthesis of the starting compound RuBP. So all the steps are being covered here, all the phases are being covered here. So if we take the example, for example, if we have to write one equation on this. So what we will write is 3 CO2 molecules plus 6 NADPH molecules plus 9 ATP will give rise to G3P plus 6 NADP plus ions and 9 ADP molecules. So this is about the Calvin cycle. The next is about how can we control the Calvin cycle. So Calvin cycle occurs during the daytime and is strictly dependent on the products of the light reaction, that is NADPH and ATP. So it will be dependent on the light products NADPH and ATP. The metabolic activity at night is mainly shifted to carbohydrate oxidation. So when the plants consume oxygen rather than producing it. So this Calvin cycle does not occur in the plants, does not occur at the night. So that the reason that it does not occur in night is because the metabolic activity in the plants, which is during the daytime, it is being shifted in the night, which is to carbohydrates oxidation. So when the plants consume oxygen rather than producing it. So we have seen that in night the plants will do the process opposite to the daytime. So this is the reason that the Calvin cycle is not in the it is not does not occur in the dark at night. So the control of Calvin cycle means the control of regulation between light and the carbon fixation cycle. So the control here means this, the light and the carbon fixation cycle, the control of regulations. It does not mean that we have to control the Calvin cycle. It is the regulation which we have to control between the light and the carbon fixation cycle. So the enzymes of Calvin cycle are very sensitive to pH. They are very sensitive to pH because the optimum pH for RuBisCO activity lies around the 8, pH 8. So in the presence of light, protons are pumped from the stroma into the lumen from the thylakoids. So thylakoids and lumen are the parts of the plants only. In the chloroplast there will be a mention of, there will be a labeling of lumen and thylakoids. So high pH and increased magnesium concentration, they activate the RuBisCO and thus Calvin cycle. So RuBisCO is also regulated by natural sugar phosphate. So this is the natural sugar phosphate which is being regulated by the RuBisCO. Then the regulation of enzymes which is involved in the RuBisCO regeneration, that is the enzymes that is involved in RuBP regeneration is fructose biphosphatase and pseudohepatulase biphosphate. They are more active at the pH of 8 and at the pH of 7 something, and they require magnesium as a cofactor for catalysis. So these are two enzymes which is required for this RuBP regeneration, and they are more active at pH 8 and requires magnesium for the as a cofactor for the catalysis. They are sensitive to the redox potential of the chloroplast stromal compartments, and the mediator molecule is the small protein thyroredoxine. So thyroredoxine is when coupled with ferrodoxin by the enzyme, that it will lead to the inhibition of the phosphofructokinase. So in plant cells, light stimulates the Calvin cycle enzymes. So this is the main point here. The light activation is being mediated by the thiodoxin. So this was all about the RuBP and its regeneration, the control of the Calvin cycle. This was all about this. So these are some of the references from where I have taken the matter and from where I have made the slides. Thank you.